All right, so let's get started. Before we go forward, just a couple of words about the exam. So that has now been linked into the grade book, so you can see your scores there. They might, depending on who you are, might have changed a little bit, only in the upward direction. I did accept an alternate answer for one of the questions after kind of looking through. So good. If that was you, if I find any others, I'll make those adjustments. But hopefully it was okay. Not our best exam, not our worst exam. The average was about a C. The median was a B. So reminder to those of you who were with me last semester, new information potentially to those of you who were not. I do allow one retake at the end of term. So how that works is our final is super short, doesn't take very long, but we got a whole 2-hour block. So you can pick whichever exam that you did the worst on originally and redo it. Just show to me that you know it by the end of the semester, and then that'll just go in there. I do take your highest attempt, so there's not a real downside to that. So that's an option if you didn't do how you wanted to. Other exam related information. So last semester we didn't have any tutors available for this class, but they have managed to find some people who agreed to tutor for A&P. So the tutoring center does now have tutors. If you want to go over some of this stuff in detail with someone who's not me, you're always welcome to come to my office hours. You can see those linked in a couple places, including the syllabus, also included in like a little QR code right next to my door. If none of those times work for you, I can try to find some alternate times in my schedule and move some stuff around. But you're welcome to come talk to me. I have an all armchair office. Those of you who have been there. I got some like squishy, cozy chairs. I usually have some kind of candy in there, so you're invited. All of you, but preferably not all at the same time, because we won't all. But those are some options, as well as your usual resources, SI, the textbook, etc. Questions about any of that at this point? Okay. And you're also welcome to come review your exam with me, so that would be a thing you could do during office hours. If you want to see like what you really get. Okay. So when we left off, we are continuing with our blood packet, but for now we're actually going to skip our second cell category. So in contrast to our red blood cells, our erythrocytes, we're going to see that we also have white blood cells, our leukocytes, which are really involved in our immune defense, right? So this is where we have our immune defense, killing off our pathogens, hopefully keeping us healthy, potentially destroying cancer cells, as well as helping us filter out the dead and injured cells. So we will touch on them, but we'll do this in more of a worksheet form that we'll go over a little bit later on. What I do want us to do all together here right now is start in on our blood clotting. Okay. Because I think, show of hands, did you see some white blood cell stuff in phys lab? Yes, no. Hands up if you, yes, during histology. No, they removed that. Okay. In that case, we may go over this in a little more detail in lecture than previously. Try to change that. Okay, good to know. All right, but we'll loop back on that. So today's topic, we're going to be getting a handle on what happens when you get injured. So just skip forward past the white blood cell stuff and then we can use that later. Right. So It's important to keep the right amount of blood in your body. So that's hemostasis, hemo referring to the blood, stasis referring to like the homeostasis, right? Keeping something at a constant state. Okay. So when you get a cut, this is a problem for your body because you've cut open capillaries for sure, maybe arterioles, hopefully not an artery, but depends on what the cut is, right? This is a problem because now we don't have a closed loop system anymore. We don't have blood just going round and round through our systemic circulation and our pulmonary circulation. We're actively losing blood. There's a problem. Your blood pressure is going to drop. You're going to pass out. You're not going to get oxygen to your cells. All sorts of bad things. So we got mechanisms to heal this real quick. So we're going to start out in this long process by having those blood vessels that get injured spasm. Then we're going to end up using our platelets, which are like little fragments of cells, essentially, form a platelet plug over the injured site. And then we're going to have the formation of a true blood clot that's going to close over any gaps until we're able to actually heal that blood vessel. So we're going to start with our damage to the blood vessel. Okay. So what happens first is that our blood vessel is going to immediately kind of constrict to minimize blood loss. So this is an intrinsic vascular response. So you can analogize it kind of to that myogenic response to a blood vessel stretching, right? We've had it clamp back down. That was one of the ways we regulated blood flow. This is another kind of automatic reaction that you have. This does also have a little bit to do with our sympathetic innervation. It's a little bit sometimes involved there. That's our first immediate response. We can just clamp down, right? So clamping down helps immediately because as we clamp down, we got a small radius, which means we got high resistance, which means we're lessening blood flow to the area, right? So you're going to have less blood loss if less blood is flowing to that region in the first place. So that's thing one. The next thing that's going to happen is that the layer of that blood vessel that's gotten cut, that's actually touching the blood, it's going to start to get sticky. All right. It's going to start to get sticky because what we want to have happen is we want to attract a bunch of platelets and we want to have them start to seal that region over, right? So the stickiness is important for this. Okay, so for this formation of the platelet plug, we will trace through what a platelet is. So the rest of our leukocytes, the white blood cells, are like over here. The platelets are these little fragmentary cells that we see all the way to the left. So sometimes they're also called thrombocytes because like thrombosis, if you ever hear that word, has to do with clotting, right? So these are cells kind of related to clotting. Yeah. So they don't have much in them. They're just like little pancake things. All right. So at that site where we injured our blood vessel, where we have this cut, we're going to start to have those platelets collect together and stick to the inside of our blood vessel in order to form what we call the platelet plug. Okay. So this platelet plug, first thing, right? You got a plug on something, you're blocking the escape of fluid, right? So we're already starting to plug over that blood loss, right? So we're keeping more of the blood in our system, okay? And this is the first step on our way to a blood clot, right? So it's not as stable as a clot, not as long lasting as the true blood clots that we'll see in a little bit, but it's our first step and it helps. So if we were looking at a platelet, like these little fragmentary cells, we would note that they're colorless, right? So they're not red like our erythrocytes were, right? They don't have a nucleus, okay? But they do have some organelles and some little granules. So granules are some of these little dots that we see in some of these white blood cells. So this includes Secretory products just means like stuff that they can give out, right? So they can secrete certain chemicals that are in those granules. So the granules are going to be like little baggies of chemicals that they're going to need to kind of signal to other platelets to tell them to also come stick here to the platelet plug and make it bigger. Okay. So you've got like 100,000 to 500,000 platelets in every milliliter of blood. So you got a bunch of them. So after our blood vessel, just quick spasms to cut down our blood flow to an area, right? We're going to be thinking about what's going on with those platelets. Okay. I'm going to do a little drawing. All right, so we're thinking about our blood vessel, right? We're going to have our site of injury. So I'm going to draw a really bad drawing of like a knife. Okay. So here's where we have our blood vessel damage. So as we damage this blood vessel, what we're doing, right, is exposing some of the layers there in that blood vessel, right? So we had, depending on what the blood vessel is, right, we always have that endothelial layer, endo for inside. And we can analogize to the epithelium, which was our skin. So kind of like almost like a skin layer, but lining the inside of a cavity, right? So we had that endothelial layer. Then we had maybe some smooth muscle, depending on what we're looking at. Not if we're looking at a capillary. Yes, if we were looking at something like an arterial. And then we had that outer layer of more fibrous tissue. So like some collagen, lots of connective tissue, maybe some elastin in that more external wall of the blood vessel. So if we got a cut into the blood vessel, now the blood can touch each of those layers, right? It can touch every part as it's escaping. Okay. So we have exposed the subendothelium, aka the layers deeper away than the endothelium. So the endothelium was that inner layer. The subendothelial layer is as we're going up through the wall of the blood vessel. Okay. So we've exposed the subendothelium. One of the things that we have in our outer layer of the blood vessel, in that connective tissue layer, is collagen. Okay. So when we have exposure of these collagen fibers, this is going to kind of trigger the action of something called von Willebrand factor, abbreviated VWF. The reason we care about this von Willebrand factor is that platelets bind to it. So when we say that we're going to end up making the inside of this blood vessel sticky, what's really going on there is that we have this von Willebrand factor activated as we have contact between the blood and the collagen that's forming part of the wall of the blood vessel. Okay. Platelets start to bind to it. So they adhere, right? Adhere really just means kind of like stick to, clamp onto, right? So platelets are going to start to adhere to that site. The platelets themselves are going to secrete chemicals that make them more sticky. Then they're going to aggregate to each other, forming this larger platelet plug. So as they start to aggregate and become a larger platelet plug, right, they're releasing these secretory products that were in the granules inside the platelet. So we have a couple things that they're secreting. One of these is something called ADP. So this is increasing that stickiness of the platelet. And this is one of the few places in physiology where we get positive feedback, right? So usually when we're thinking about feedback, we're thinking about negative feedback. We're thinking about, we have a change in one direction, we don't like it, negative feedback pulls us back to normal, right? Whether that factor went up or down, we're just trying to get to our normal stable state. What happens in our positive feedback, right, is we start to have a change. And then we have a snowball effect, right? Like just that little bit of change makes that factor change more and more and more and more. More positive feedback is increasing whatever is happening, right? Okay, so when we have ADP released, right, we started out with a region that's a little bit sticky, right? Then the platelets secrete ADP, making this whole collection of platelets more and more sticky, right? So they're getting stickier and stickier and stickier as we're releasing more and more ADP. We're also releasing serotonin and epinephrine, both of which have the effect of causing vasoconstriction. So usually when we think about serotonin, we think, at least in the general world, right? We think about happiness, we think about chemicals in the brain. But serotonin actually has a lot of roles in your body, including out in your body. So one of the roles of serotonin is to help those blood vessels constrict when you have an injury. Epinephrine causes vasoconstriction as well, and is binding to those alpha receptors in that smooth muscle, potentially in the walls of blood vessels. We saw how those alpha receptors cause vasoconstriction. There are other chemicals that the platelets are releasing as well that help us form our blood clots. So coagulation is just really another word for clot. So we're gonna also start out our clotting process with some of these secretory products from the platelets. Another chemical that they're releasing is something called thromboxane A2. And this is also going to have the effect of creating a positive feedback loop where we see more and more collection of platelets, eventually clotting in this region. Now, platelet plugs are great if you have an injury. They're not great if you don't have an injury, right? Like clotting problems, when we think about clotting problems, we might think about you can't form clots, and so you bleed too much. You got a cut, like hemophilia. But more often when we talk about clotting problems, we're talking about like too much clot formation, right? That's how we end up with things like strokes and heart attacks, right? It increases the risk. If you're forming clots where you shouldn't, they eventually might break off and get stuck in smaller blood vessels somewhere. They get stuck somewhere important. That's bad for you, right? So we want to prevent our platelets from getting stickier and stickier and stickier and having that plug spread out and start to cover over healthy tissue, because we want healthy tissue to stay free of clots in general, right? We want healthy tissue to be nice and smooth, so blood flows past nice and smooth, no problems, no extra clots, no clots breaking off and getting stuck places. Okay. So next to our injury site in our healthy tissue, we're gonna have a different thing going on, right? I'm just gonna draw a gap here. So this gap is gonna be that injury site. Right. So I'll mark off the edges, right? So here's where we have von Willebrand factor involved. We have explosion of those collagen fibers. Our platelet plug is forming here, right? But as it's getting bigger, you could imagine, right? It doesn't know directions necessarily, right? So you could have it spread out way far to the sides and just create like a whole internal layer all around the inside of the blood vessel, but we don't want that to happen, okay? So we have ways to prevent that. All right, so the healthy cells that haven't been injured, right? So if we pick out a cell like here, right? So we have a healthy endothelial cell. What a healthy endothelial cell is going to do is it's going to be secreting prostacyclin, and it's going to be secreting nitric oxide, which are going to help us inhibit this aggregation of the platelets. They're going to help us stop the spread of the platelet plug. Right. So here we have the pictures of this process. They're going to be better than my pictures. So let's trace through what we're looking at here. Okay. So here we have those cell layers, right? So we've got a healthy endothelial cell here. Neighboring it, we have an injured endothelial cell, right? It's cut off We can see that it doesn't have this nucleus, right? We're missing part of it at that injury site. Then we can see that smooth muscle layer, right? And we're also going to have exposed collagen fibers kind of along the outer edges of this. So we just call the whole region the subendothelial layer, right? Because we may or may not have a bunch of smooth muscle, right? But we will definitely have at least some collagen. Okay. All right. So on those exposed collagen fibers, right? This is where we first start to have the platelets adhere, right? So they're going to adhere to those exposed collagen fibers because von Willebrand fracture is telling them to, right? So this area got sticky a little bit. We have the platelets adhere. So that's number one. Okay. As those platelets collect, stuck to those collagen fibers, they're giving out their secretions, getting stickier, 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 their positive feedback loop, which is what's causing the platelet aggregation. So aggregation, and aggregation means a bunch of things collected together. Right? So aggregating, we're just talking about collecting a large group of platelets, sticking them together into the plug. So this platelet aggregation part is basically step 2. Now we can see our positive feedback loops here, right? Going along, positive feedback loop is why we have a plus, right? So we can see that we have that thromboxane A2 being secreted by the platelets, right? So this is part of that positive feedback loop. Okay? We also have the secretion of ADP, right? Also part of the feedback loop. And in fact, we can see that in actuality, these two interact, right? The thromboxane A2 is also helping us ultimately increase that ADP. And the total effect is that this platelet plug is getting stickier and more platelets are collecting together. So that platelet plug formation and growth through positive feedback will be kind of like step 3 here. So what's happening at neighboring regions, right? So you could imagine this cell, right? Or you could imagine a cell over here, which is what we're looking at when we're looking at B. We're looking at why this jumble of platelets isn't spreading out to the side as we have more and more positive feedback. It's getting stickier and stickier, should be getting bigger and bigger. We want to know why it's not totally closing over our entire blood vessel. So this is where we have that secretion prostacyclin and the nitric oxide, which prevent this aggregation, right? So you could imagine them as being like minus on this process, right? So they would create like negative feedback. So the platelet plug is our first part, right? So we need the platelets to adhere because of that exposure of the collagen fibers acting on Willebrand factor, making that cut sticky. They adhere, stick to it, right? Then we get more and more platelets. They're aggregating, step 2, forming that plug. Positive feedback loop, making that plug bigger and bigger. That's our sort of first part on our way to a blood clot. Okay. Actually, I guess the first part was the blood vessel clamping down, right? But the first change that we see to the walls of the tissue is the plug. So the blood clot itself is actually going to be made of a nice big mesh of something called fibrin. Okay, so what we're trying to do over that cut is create like a screen kind of, right? We want to make a nice web, right? I want to make a nice big stable, just closing it over, right? We don't want a big jumble of platelets just kind of all in the way. we're going to close this over with a mesh. Okay. So a blood clot is going to happen through coagulation, just the same thing, fancy word. Okay. So we're converting components of the blood into this solid gel. If we're calling it a clot, or again, if we want to be fancier, a thrombus. Okay. Same thing again. This is happening around the platelet plug. So platelet plug is starting off blocking that area. The clot is then coming in, helping hand at the same point. Okay. This is our dominant hemostatic defense mechanism. Okay, this is the main way that we're stopping blood escaping. Okay. Now, there are a lot of different chemicals and signals involved. So we're going to kind of make sure that we're focusing in on the most important parts and then kind of adding details and zooming out. Okay. So the most important parts of the formation of the blood clot, we're trying to get to this stable fibrin mesh. Okay. And the way we do that is we have a chemical called fibrinogen. It's just like kind of always in your blood. It's there the whole time. And it's our precursor to this molecule fibrin. So actually, anytime in this course that you see this like inogen ending on a word, that should be your signal that this is some kind of precursor molecule. We're going to see this again when we talk about like enzymes that get activated to break down proteins in your gut. This inogen ending means something. So fibrinogen is kind of like our inactive kind of form, right? Starts by making fibrin. It's a little loosey-goosey. It's not very structured, right? It's not in this mesh form yet. Eventually, it gets stabilized into this stable fibrin mesh. And fibrinogen, loose fibrin, stable fibrin in a mesh. So the clot itself is made of fibrin. Oh, I should have made this a Slido question. We'll practice these next time. Okay. So when we're talking about pathways, right, they're going to get intense. So what we want to make sure that we're doing, right, don't lose sight of our goal. Our goal is to convert fibrinogen into loose fibrin into stable fibrin, right? So all the rest of the pathways that we're going to see, we're just trying to get down to this level, right? So we're just trying to figure out, why does the fibrinogen become loose fibrin? Why does the loose fibrin become stable fibrin? Okay. Now, in your bloodstream, you've got fibrinogen, you've got this precursor molecule that's eventually going to become fibrin when it's activated. You also have a ton of clotting factors. Okay. They all have numbers. The numbers are not in order. They're got numbered in the order I think that they were discovered rather than like the order in which they need to act, which is frustrating. So that's a PSA. Don't get put off by those numbers. Okay. The ones that are most important, mostly also like have names, right? So here we can see prothrombin and thrombin have their own names, but this is actually clotting factor 2 and clotting factor 2A, okay? But some of them will only be referred to as their numbers. Right. So we'll trace through this a couple of times in a couple of ways. But this is like the smaller version of this picture. Okay. So we're starting out with this fibrinogen. Okay. And we have some intrinsic pathways, so like some local pathways that are going to help us convert that fibrinogen into the loose fibrin to start with. Okay. So we've got cascades of clotting factors. Okay. Most directly, what converts that fibrinogen to fibrin is thrombin. Okay. So we can see that thromb part of this word. A thrombus is a clot. Thrombin is the clotting factor that turns our precursor fibrinogen into fibrin, which is a clot. Okay. So thrombin is responsible there directly for turning fibrinogen into fibrin. It's also indirectly responsible for the stabilization of this network. So we can see here that thrombin needs a little help, but another thing that thrombin is doing is activating this clotting factor 13. Right, so we went from inactive 13 to 13A, A for active. Okay. And that's the molecule that's directly stabilizing stabilizing the fibrin. Okay. So zooming back out. Okay, so we needed to turn fibrinogen into loose fibrin. Thrombin does that. Then we need to turn loose fibrin into stable fibrin. We've already got the thrombin acting. The thrombin is also going to help us stabilize. We just got an intermediary step. The way that thrombin allows us to stabilize this stuff is that we've got this helper molecule, clotting factor 13 in there. Okay. So then we can zoom out a little more. Okay. So we also need to have the thrombin in the first place, right? So thrombin is going to help us make this mesh out of fibrin, but where did the thrombin come from? Thrombin started out as prothrombin. So pro, think kind of like pre, right? So this is our precursor molecule for the thrombin. Okay. All of that got turned into thrombin by the action of factor X. Okay, so for our intrinsic pathway, the way we would think through it, we got a cut. Right, so our intrinsic pathway immediately, like from the blood vessel itself, right? We're going to have signals that give us an active factor X, right? We're going to activate factor X, factor 10, okay? Once factor 10 is present, it turns prothrombin into thrombin, right? So it's activating another clotting factor. That thrombin then turns our fibrinogen into loose fibrin, starting out a kind of unstable clot. And the thrombin activates another factor, also stabilizing that fibrin into its kind of final mesh form. This is also a positive feedback loop as thrombin activates more and more of this factor. So, questions about this part of the pathway? Right. So we're zooming out a little more. All right. So here is what we were looking at just now for our intrinsic stuff. The part that we're focusing most on is still at the bottom, right? Our concern is still just we need to turn fibrinogen into a stable blood clot. We've got this middle step where we have loose fibrin. Okay? So now we know that thrombin is really important for this because thrombin activates the fibrinogen, and it also, through an intermediary, helps us stabilize the mesh. We know that we got thrombin around in its active form because of this factor 10. So that leads us to the question of, okay, but why do we have factor 10? So we're not going to go through all of these steps. So our answer is going to be that we activate that factor 10, which is this kind of like central point here where we have multiple pathways joining all together, either through contact with that collagen. So just like that formation of the platelet plug happened because we had that collagen in the subendothelial layer exposed, right? The same thing, it's kind of triggering this fibrin clot to form, right? So we've got lots of numbered factors here. We're not going to learn them, but it's that contact with collagen that starts this process off here. So that's our intrinsic pathway because collagen itself is part of the blood vessel. Collagen is part of the blood vessel. So this is an intrinsic pathway. It's local property of the blood vessel itself. Our extrinsic pathway, when we say extrinsic, we're thinking like external. The external stuff that we're thinking about is the tissue surrounding that blood vessel. So like, if we're thinking about our blood vessel with its injury, right, we got the gap, right, our collagen fibers are somewhere here, right? So that's helping us trigger both that platelet plug formation as well as this fibrin clot formation, okay? But your blood vessels aren't like the surface of your body, right? You shouldn't have any exposed blood vessels touching the air right now. So if we are getting an injury, right, we are also damaging the tissue surrounding on our way there, right? So we've also got cells like skin cells, fat cells, whatever, around that blood vessel, organ cells, pancreatitis, right? Okay. So we have surrounding tissue that also gets injured on the way to injuring that blood vessel. So that is what is responsible for this extrinsic pathway. Okay. So we also have these tissues secreting their own signals that activate this factor 10 and lead us into the rest of this cascade. So that's really why we're focusing on the common process, right? So all we need to know separately about the intrinsic versus extrinsic pathway. So the intrinsic pathway has to do with that blood vessel wall itself. It's getting triggered by damage to the blood vessel wall. But that extrinsic pathway is happening because of damage to the surrounding tissue, which also causes the activation, the secretion of factors related to clotting, all ultimately cascading all the way down to the creation of this stable fibrin mesh. We'll practice this a couple of times over the next couple of days because I know it's kind of a lot. Here we see that intrinsic pathway leading us all the way down to factor 10. Active factor 10 is what really kicks off the whole thrombin, fibrinogen, fibrin deal. We have our extrinsic pathway also leading us to that factor 10, activating it. And so together, factor 10 turns prothrombin into thrombin. Thrombin activating that fibrinogen, turning it into loose fibrin. And then thrombin indirectly is stabilizing that thrombin meshwork. So in terms of focusing, as you're going through this and explaining it to yourself, right, we're keeping this goal in mind, right? Our goal is just to turn fibrinogen into loose fibrin, into stable fibrin. But if we're learning this pathway, right, first step is we've got to activate factor 10, however we do that. Factor 10 turns prothrombin into thrombin. Thrombin turns fibrinogen into loose fibrin. And then it's also indirectly stabilizing that blood clot. That's the order I kind of recommend you think about this. Questions about this cascade initially now? You know, you'll probably need to let it settle a little bit before you form. Good question. For greater questions. not immediately. Okay. Okay. So as we're forming these blood clots, we have these clotting factors in your blood all the time because the liver is making them. So your liver makes clotting factors all the time, but in their inactive form, because you only want to have a blood clot when you need it, right? You don't want clots forming if you don't need them, because they're dangerous for your body. So they're inactive until they get activated by this whole cascading effect of other clotting factors, which happen in response to the injury, either through that indirect pathway where it's actual contact with that collagen that starts off our cascade, or through injury to the surrounding tissue when we have tissue factors activating that same kind of final part of our cascade. So these are in your blood. When you donate plasma, you can donate it with platelets in there, right? But sometimes we're also going to want just plasma without these clotting factors in it. So when we refer to plasma without clotting factors, so without these chemicals, we refer to it as serum. So if you hear blood serum, what we're saying is that we've got plasma, so we've got that watery fluid, we've got some of the things that are dissolved in that fluid, right? Not red blood cells, but like yes to ions, yes to maybe some of the proteins, but specifically we don't want to have these clotting factors in there. Okay. We do have some other necessary substances for clots to form that aren't clotting factors. So one is calcium, and sometimes they give it a factor number, factor 4, but it is also just the ion calcium. We also have a platelet factor that's important for this process. Platelet factor 3 is also involved in these overall cascades, but we're not going to zoom in too much on those effects because just the basics of the clotting cascade are kind of tricky to get your mind around when you first see it in the first place. But that's why we see these things here, right? So you can see some of those other components that we need in order for this clot formation to happen on those lines. So here's that calcium and that platelet factor interact. Which is also why that platelet plug formation has to happen first. Just like we have things that stop our platelet plugs from getting too big and spreading out to healthy tissue, we also have things that can limit clot formation. So like this actual fibrin clot. So we refer to them as anticoagulants because coagulation is clotting, anti, the opposite of. Okay. So we have some proteins in your plasma and on the surface of healthy endothelial cells that is supposed to stop your blood from forming clots if you don't have an injury. Okay. So one is coming from the tissue, kind of. So we have surrounding tissue that can trigger blood clot formation, right, when it gets injured, okay? But healthy cells, healthy endothelial cells, are going to inhibit that tissue factor, right? So if your blood vessel isn't damaged, sometimes you'll have like a bruise, right? So you can imagine a situation in which your tissue is injured and might be sending off signals of that injury, but maybe your blood vessel isn't actually damaged, right? You wouldn't want it to form a clot, right? So those healthy endothelial cells in your blood vessel inhibit that extrinsic pathway by inhibiting the tissue factor. Another anticoagulant that you have in your body, also, again, secreted by healthy endothelial cells, right? So anytime we're talking about stopping clotting, right, we're thinking about things that healthy cells are going to secrete. Right? So healthy cells, healthy endothelial cells also secrete something called thrombomodulin. So think like modulating thrombin modulating a clot, right? So we're like affecting the clot, we're changing it a little bit. What's happening is that this thrombomodulin, this anticoagulant molecule, binds to thrombin. We know that thrombin is principally responsible for turning fibrinogen into fibrin, as well as indirectly responsible for stabilizing that fibrin mesh. Okay, so if we can affect the thrombin, then we can affect that clot formation. Okay. So when it binds to thrombin, it forms a complex that activates protein C, and protein C like kind of directly helps us stop that coagulation from continuing. So here's that big picture, and just so you know, this is like how you can imagine it kind of happening in your bloodstream, right? So they're always just kind of there, made by your liver. And then we have those varieties of factors getting activated, forming this clot that also happens to trap red blood cells in it, forming some of the body of this clot. We've also got some platelets going on, forming a platelet plug, lots and lots of signaling, secretions, all sorts of things. The process is complex. We want to focus on our goals first, and then kind of add on details once we get down that solid basis. Okay, so we will stop there and review, do a little kind of quizzing ourselves at the beginning.